Today we're going to be talking about rates of change, and there are examples of rates of change everywhere. So the first one that you maybe are most likely to think of is the speed of driving a car. So we always are thinking about our speed in miles per hour. And miles is our change in position. And then the hours is the change in time. So if you're going 35 miles per hour, it takes you 1 hour to go 35 miles or change your position by 35 miles. Another currently very relevant example of rates of change is inflation. We're talking about inflation all the time in this economy. And the inflation can be measured as the change in cost over the change in time. So this could be something like the change in the cost of groceries over the last year or something like that. And what both of these have in common is that this rate of change is some change in output divided by the change in input. And we should also, in this biology class, talk about some biological examples of rates of change. So the first one, and one that you are probably, again, very familiar with, is the spread of the disease, a disease. And we're going to talk about COVID, because what else would we talk about? And so here, what is being measured is the rate of spread, which we often quantify as change in the number of cases over some change in time. And so what this ends up looking like is that, like, the number of COVID cases per day. And this is data that you probably used to see everywhere, but this is the most recent data as of August 11th. On the number of cases of COVID per day, as found on Google. Another example of rate of change is the metabolism of a drug. And so the rate of metabolism is the change in drug concentration over the change in time. And an example of this is the metabolism of caffeine. And in this example, what we are looking at is the change in the concentration of caffeine over some period of time. In this example here, it is over minutes. So let's talk about some notation for the rate of change. So we say that the rate of change is the change in output over the change in input. And often in math, the change is represented by a delta. So this would be the delta in output over the delta in input. And at some instantaneous point, so at a single point, this is often abbreviated to D. So actually an example of this would be for COVID, the change in cases, delta cases over the change in time, which we could, if we say that cases is equal to C, we could do this as delta C over delta T, or dc dt at a single point. we go back to our caffeine example, we were again looking at change over time. So the delta in caffeine concentration over the change in time. If we say that caffeine concentration is equal to C, and we'll pretend this is capital C, that one was lowercase c, just to differentiate between cases and caffeine. Again, we would be looking at delta C over delta T, or at a single point in time, dc dt. So let's talk about how these rates of change are visualized. In this COVID example, the x-axis is time and the y-axis is cases per day. So cases per day is already a rate. So if we wanted to get the total number of cases, we would be looking at all of the cases, which would be area under the curve. So to get cases from this example, we would get area under the curve. Because the number of COVID cases is always increasing, just the rate at which it's increasing varies, and that rate is what is plotted here. In our caffeine example, we are actually looking at the total amount of caffeine over time. So here, what the rate is actually going to be the slope of the line. So out at this point here, there's the slope, or dc dt is going to be, or dc dt is going to be the slope of this line here. Over here, early on, the slope, or dc dt, will be positive very fast. So while the amount of caffeine is always positive, always above zero, the rate changes from being positive to negative. So the rate at which the amount of caffeine in the bloodstream is increasing is very fast and positive up until it reaches its maximum. So if we were to over here plot time and plot the rate dc dt, where this line here is 0. I'll go ahead and erase this line here, so you know that those are the axes. And so up until some point, it's going to be positive, but it's decreasing, right? So it increases really rapidly here, a little slower here, and then much slower here. So the rate might look something like this up until this inflection point, which I will mark with a star. We'll also say this is the star. And then the amount of caffeine in the blood is decreasing, and so the rate is negative. Right, so there's two ways to visualize rates of change. One is to plot the rate, and one is also to plot the total amount of whatever it is you're looking at, and then the slope of the line is the rate. So another biological example that I want you to think through is the levels of blood glucose over time. and the rate of change in blood glucose concentration over time. Right, so just as a refresher, the blood glucose is the amount of glucose or sugar in the blood. It is one of the things that diabetics are frequently measuring. And in a person without diabetes, the blood sugar should stay between 80 and 120. So kind of within this range. And you all learned in 2200 or 2220 how blood sugar is regulated by insulin. And just in case you forgot, when blood sugar levels are high, beta cells in the pancreas release insulin, and that allows glucose to enter the cell and then thus leaving the blood going into the cell by activating the glucose transporter, which brings glucose into the cell. And this is a way that blood sugar levels are regulated. So what I want you to do is to think through this system and draw out, however you see fit, this process and how it is regulating the amount of glucose in the blood, or the concentration of glucose in the blood. And then as your second part of that question, just think about what would happen if no insulin was produced, as is the case with type 2 diabetes. And we will talk about this in class when we meet next.